Hi everyone, my name's Dr. Elizabeth Duncan and I'm a lecturer in zoology within the School of Biology here at the University of Leeds. Thank you for coming to my taster lecture. Today I'm going to take this opportunity to tell you a little bit about my research. But before we get started, the thing that people often notice first about me is that I'm not a native Yorkshire woman. I'm from New Zealand and I moved to the University of Leeds 5 years ago to start my research lab and begin my career as a lecturer. And one of the reasons that I chose the University of Leeds is that we believe in research-led teaching. So our students here are lectured by experts in their research field. Also, as an undergraduate, you have an opportunity to get involved with our research. And in my talk today, I'm going to talk about a couple of examples where undergraduates have contributed to research projects that I'm running. As a biologist, one thing that has always fascinated me is the diversity of form and function seen amongst plants and animals that surround us. These plants and animals are uniquely adapted to their environment. One example can be seen when comparing cows with giraffes. So cows and giraffes are actually quite closely related species. But giraffes evolved in Africa, and they have fringed tails to flick off insects from their flanks and long necks so that they can access food high up in the trees, which is an environment that cows don't experience. So these animals look different, they're adapted differently to their environment. This idea that animals are adapted to their environment was formalized by Charles Darwin in a really famous book called On the Origin of Species. And this book ends with a wonderful quote. "From so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved." And this quote encapsulates what I study because I study evolutionary biology. So how did I know I wanted to study evolutionary biology? Well, actually, I was sat in a lecture theatre. I was an undergraduate student in a second-year genetics lecture, and my genetics lecturer said to us that the phenotype of an animal, the way that it looks, the way that it behaves and its biochemistry, is entirely dependent on interactions between its genes or its DNA and the environment. And I was intrigued with this idea, and I went up to the lecturer afterwards. Now, I was a shy undergraduate, but I went up to the lecturer afterwards and I asked them actually how the environment can have such a profound effect on the way an animal looks and behaves. And at the time he said, "We don't know." Now, using very stereotyped examples of gene-environment interactions, we're now beginning to understand how this works. And actually, this is what I'm going to talk to you about today as an example of phenotypic plasticity. So why are we so interested in the phenotype of an animal? Well, actually, it's the phenotype of the animal that evolution acts on. So plants and animals that are best adapted to their environment, survive the longest and reproduce the most, are the ones that we see surviving around us today. So this diversity of form and function that we see, these endless forms most beautiful, are evolutionarily successful. They're adapted to their environment and they've all survived to be successful and reproduce. In my research, I study very stereotyped examples of gene-environment interactions in insects. And there's lots of reasons to study insects. Firstly, they're really important. And so two of the insects that I study, one is a pest species, and this is the pea aphid. And I'm not going to talk much about the pea aphid today. But when I tell people I work on the pea aphid, people most often ask me, how do I get rid of them from my roses? Now, I'm not a gardener, so I can't tell you how to do that, but I am going to tell you something really cool about aphids that will hopefully give you a new appreciation for them. Aphids change the way they reproduce depending on the season. So in summer, aphids reproduce without having sex. So the aphid that I'm showing on the screen there on the left-hand side is an adult female aphid, and you can just see these dark spots in her abdomen. Those are actually the eyes of her offspring inside her. So she gives birth to live young, and those live young already have their offspring developing inside them. They're like the Russian dolls of the insect world. But when the temperature changes and the day length decreases, the aphid senses this change in the environment, and she changes the development of her offspring to give birth to females that reproduce sexually. So they mate with a male, and instead of giving birth to live young, they actually lay eggs. And those eggs can overwinter, hatching in the summer into live aphids. And that's why aphids are such successful pests, because they reproduce without having sex. Now that's all I'm going to say about aphids today, but I think they're very cool. And in fact, when people ask me what my favorite insect is, I often say aphids because they don't bite or sting. But while aphids may be my favorite insect I work on, the honeybee is the most important insect that I work on. So the honeybee is a really important pollinator species. And pollination is an example of an ecosystem service. It's a service that these insects provide that is of benefit to us and to the ecosystem. And pollination is really important when we're thinking about crop pollination and food security, but also important for maintaining floral biodiversity in the environment. So I use honeybees to study gene-environment interactions. So how is the environment having such a big effect on the phenotype of the honeybee, the way that it looks and the way that it behaves? A honeybee colony can be made up of 100,000 individuals. And almost all of those individuals are females. Males actually play a very small part in honeybee societies. But of those 100,000 individuals, there is only one female that reproduces, and that's the queen, and she's shown at the bottom of the screen. Now the queen can lay 2,000 eggs a day. She is an egg-laying machine. All of the rest of the bees in the colony are worker bees. They're also female, but they can't reproduce. And those worker bees, it's their job to look after the queen and to look after all of those eggs and larvae that she has laid. So to look after all of her offspring. So workers have relatively short lives. They live for about 4 to 6 weeks. They have pollen baskets on their hind legs that they use to pack the pollen in when they're out foraging, and they have small inactive ovaries. Queens, on the other hand, they develop much more quickly, they're bigger, and they live longer, up to 4 years in fact. They have jagged mandibles, which are their mouth parts, and she produces different pheromones, so she smells different. And one of the pheromones she produces is called queen mandibular pheromone. And I'm going to talk about that pheromone a bit later today. The queen also has a spermatheca, she can mate, and she has large active ovaries, which is how she lays 2,000 eggs a day. Now, if a queen and worker bee were sat side by side on the table, it would be very easy to suggest that they're different species. They look quite different. The queen is bigger, and she's got these jagged mandibles, but actually, not only are they the same species, but they're largely derived from the same DNA. So how do we get a queen and a worker, these animals with 2 different physiologies and 2 different phenotypes, developing from the same DNA? The answer is this white substance that you can see on the right-hand side of the screen. In these cells, you can see a honeybee larvae sitting in the middle of each of these cells, surrounded by a white substance. This white substance is called royal jelly, and it's made in the heads of worker bees. They then feed this substance to this larvae. And this substance, this royal jelly, is sufficient to trigger queen development. So a honeybee queen will lay an egg. After a few days, that hatches into a larvae. If the worker bees want to make a new queen, they feed that larvae royal jelly. And this royal jelly substance triggers queen development and these different morphologies and behaviors that we see. So people often think that it's the queen that's in charge of the honeybee colony because she lives for a long time. But actually, if the workers aren't happy with their queen for any reason, if she's not laying enough eggs, they simply make another one. They choose a larvae and feed it royal jelly, and they supersede the queen. So this nutritional stimulus is sufficient to trigger queen development in a honeybee larvae. It doesn't matter about the DNA, it's this environmental signal that acts on the DNA to generate these different phenotypes. And this is what's called phenotypic plasticity. Now, this effect of nutrition is also seen in a range of different animals, including in us, in humans. So we know that in humans, what your mother ate when she was pregnant with you determines your lifelong susceptibility to adult onset diseases like type 2 diabetes and obesity. So the environmental signal that you received when you were developing in your mother's womb 30 or 40 years ago can determine your likelihood of getting these adult onset diseases later in life. So there's a lot of interest in trying to understand actually how animals sense their environment and how this has such a profound impact on phenotype and disease susceptibility. So I said on the previous slide that queen bees have large active ovaries. And in the photograph at the bottom on the left-hand side, you can see a picture of a queen ovary. These ovaries are quite large. They're about the size of your little fingernail, and she has 2 of these ovaries inside her abdomen. In insects, ovaries are made up of long strings of developing eggs called ovarioles, and you can think about these as being conveyor belts for egg production. At one end of the conveyor belt you've got immature eggs, and at the other end you've got mature eggs that are ready to be laid. The honeybee queen has about 200 of these conveyor belts for each ovary, and that's how she's able to produce more than 2,000 eggs per day. In contrast though, because they weren't fed royal jelly during development, worker bees actually have very small ovaries by comparison. They only have about 6 to 12 of these conveyor belts per ovary. And when we compare the size, where the queen ovary is the size of your little fingernail, the worker ovaries are about the width of a piece of cotton, and so very difficult to find when you're dissecting the workers. It's quite a skill, I tell you. What's remarkable though is that worker bees can reproduce. So if the queen is lost or removed from the hive, the workers sense that change in the environment, and that change in the environment affects their ovary. And so they go from having these small ovaries that are as thin as a piece of cotton to ovaries that are much larger, and at the bottom of the screen there you can see that they produce cotton to having larger ovaries that produce mature eggs. Now these are not as big as a queen ovary still, because she still has a smaller number of conveyor belts, but she can produce mature eggs. Now due to a quirk of genetics in honeybees, and in fact in all Hymenoptera, so all bees, ants and wasps, these eggs only produce males. So honeybee workers can't mate, so they can't fertilize their eggs. But in Hymenoptera, eggs that are unfertilized actually produce male offspring. And so I've shown you at the bottom here a picture of a male honeybee. I think he's gorgeous. He's got large eyes and a large thorax. He looks a little bit like a teddy bear. And his purpose is to mate with the queen. So he has those large eyes so he can find the queen, and he can't sting. So honeybee workers can produce male offspring. So what stops honeybee workers from reproducing? Well, it turns out when a queen is present in the colony, she produces a bunch of different chemicals called pheromones, and those pheromones are passed throughout the colony between the workers. One of those pheromones, called queen mandibular pheromone, is important for repressing reproduction in worker bees. This queen mandibular pheromone is an environmental signal, and when the queen is lost from the hive, it's that signal that the bees are responding to. So this is the system that I'm using to try and understand gene-environment interactions. And the three questions that I'm trying to answer in my research that I think are really important in terms of understanding how gene-environment interactions happen is, first of all, to understand how the bees actually sense this environmental stimulus. How do they know whether the queen is there or not? So how do they sense that queen pheromone? And we think that the brain and antennae are really important for that. The second question is, we know that the ovary is affected by this environmental stimulus. So how is the signal transmitted between the brain and the ovary? And finally, how does this environmental signal change the biology and the behavior of the worker bee so that the ovaries become active and the bees start to lay eggs? There's also some quite substantial behavioral differences between workers and laying workers as well. So to address these really important questions, I use a variety of techniques and a variety of resources, but probably the most important resource is the School of Biology Research Apiary, which is located just outside the biology building at the University of Leeds. Inside the apiary at the moment we have 10 honeybee colonies, and each colony is made up of multiple frames like the one you can see in the photograph here. Most of the bees on this frame are worker bees. You can just see the queen disappearing away over to the other side. I put a little sun over her head so that you can see her really easily. The worker bees use this frame to store honey and to store pollen, and also the queen lays her eggs in this frame. Having this research apiary at the University of Leeds means that actually we can really easily manipulate the environment that these bees are experiencing. So we can remove a queen bee from a colony, or we can put a queen bee in and manipulate that environmental effect to see what impact that has on the genes and also the biology and behaviour of the worker bees. So to do this, I use a variety of techniques, some of them in the laboratory and some of them in the field at the apiary. We do some behavioural assays to look at how individuals are responding to the queen. We look at individual differences in how they respond to various stimuli, including the queen. We use molecular biology to look at gene expression and how that changes when the queen is removed from the colony, what changes in how the genes are used in the brain and the ovary of these bees. We do functional studies, and we also use microscopy to look in very close detail at what's happening in the ovary as the ovary responds to the loss of the queen, and we use genetics. So all of these techniques we use to address these three questions about how the environment has such a profound impact on the way that honeybee workers look and the way that they behave. But this is essentially what we call a fundamental science question. So we're trying to find out something fundamental about how biology works using this approach. But honeybees also have a real-world impact as well. In fact, much of the research that happens within the School of Biology here at the University of Leeds has real-world impact. And it's important to us as scientists that our science is having a positive influence on the world around us. In terms of honeybees, they're really important for pollination. And on the screen, I've just put a handful of crops that require pollination by animals like the honeybee. My favourite is at the bottom left-hand side of the screen. This is a coffee plant. Like a typical New Zealander, I'm very, very fond of a flat white. So a lot of our crops require pollination by animals, including the honeybee, and the honeybee is the most important commercial pollinator species. However, you might have seen headlines in the newspapers like insect apocalypse or insect decline. Globally, we see insect numbers dropping, and this includes bees. There are lots of reasons that insects are in decline. Most of them have to do with things like habitat fragmentation. So actually there's less space for them to inhabit and to find food than there was 50 years ago or 100 years ago. But also we see increasing use of chemicals in farming, and in particular pesticides. So pesticides have a negative impact on all insect species. But what we're finding in honeybees in particular, that it's not as simple as these pesticides just simply kill the bees. So what we're finding is that some of these pesticides have sublethal effects. So the concentrations that are used in agriculture and found in the environment, it may not kill the bees, but it does have a negative impact on them. So as part of their normal function, honeybee workers can forage for up to 5 kilometres away from their hive. So they go out looking for flowers to collect the pollen and collect the nectar and bring it back to the hive. This requires actually very good cognitive skills. So the bees have to be able to navigate to where they want to go, they have to be able to remember how to get back home, and they have to have very good locomotor skills. What we're finding is that pesticides at even quite small concentrations interfere with this, and that this has a negative impact on the entire colony. Now, the project that I'm going to describe to you today is actually done by undergraduate students. So for several years, I've had undergraduate students in my lab working on the sublethal effects of pesticides on honeybees. Now, as an undergraduate student here at the University of Leeds, in your third year, you have the opportunity to work on an integrated research project. And you have the choice of whether that is a lab-based project or a field-based project or a data analysis project. So there's a lot of scope. And in fact, last year, there were over 100 projects you could choose from in your third year. But what's important is that you spend 10 weeks working on a real research project with an academic like myself. And so I host students in the lab, and they've been working on these sublethal effects of pesticides. And this is just an example of one of their results. So in this assay, we were looking at the effects of a pesticide exposure, in this case a pesticide called imidacloprid, which is a neonicotinoid pesticide, and those are the ones that have been in the papers a lot over the last 5 years or so. Now we were looking at the effect that this pesticide has on activity in the bees. You can see in the light blue bar on the graph, this is the control. So this is the activity that we see in untreated bees. In the darker blue bars on the right hand side of that graph, these are bees that have been exposed to pesticide. And you can see that actually even at relatively small doses, we see a significant increase in the activity of the bees. Now, this is potentially a bad thing because it means that they're using energy more than bees that have not been exposed to pesticide. So this has a significant impact on the lifespan of the bees. And this research project was carried out by 2 research students in the lab, Kelly Park and Sarah Lonsdale, who are both fantastic students to have in the lab. But there are other opportunities to get involved in research projects and pick up practical research skills during the course of your degree. One example is the Mediterranean Ecology Field Course, which is a second-year field course and is compulsory for ecology students but optional for other students. During this field course, we take students to southern Spain into a little town called Sorbas, which is just out of Almeria. And we take students to this area because actually this is a dry, arid region, and the ecology of this region is very similar to what we expect most of the Mediterranean basin to be like as a result of climate change. We're generally out in the field for about 10 days in total, during which we get 6 to 8 days of field work. And the students work in groups of about 4, and they work with an academic to design and carry out a research project during this time. This is a really great hands-on learning opportunity about the flora and fauna of the Mediterranean region. Now on the slide here, I've just got a couple of pictures showing what the landscape is like at Ura. Ura is the field station. It's just the white building in the far distance in the top photograph, and you can see that it's very arid, a semi-desert-like kind of ecosystem. So I just want to really quickly talk about a couple of projects at undergrad. So the second project I want to talk about also stems from my work in honeybees. Because honeybees live in big colonies with lots of individuals, diseases get transmitted really easily between individuals within a colony. One of the most common diseases is a disease called nosema. And nosema is a microsporidial infection, and it causes diarrhea. Now what that means is that if a honeybee visits a flower and the honeybee has nosema, it's really easy for that honeybee to leave nosema behind on the flower, and then when the next bee visits, it might be a solitary bee or a bumblebee, it can then pick up the infection from honeybees. So we wanted to determine actually how common it was for nosema to be detected in honeybees around Spain. Now this parasite can be detected by looking down a microscope, but actually that's not very accurate. And so what we did is we bought some portable molecular biology equipment that we bring to the field. And you can see a photograph here of my students using this molecular biology equipment. This means that we can use a really sensitive technique called PCR to detect whether we find nosema DNA in the guts of honeybee species. And you can see some of the results on the right hand side here. This is a gel, and where you see a white band, that's the presence of DNA. The band at the very bottom of the gel tells us that all of our experiments actually worked, that we had honeybee DNA present, but we didn't find any bands for nosema. So there was no nosema DNA in any of our samples. This is really good news for the local bees because we didn't detect any disease transfer between honeybees and solitary bees. And if you're interested in reading a little bit more about it, the company that we bought this portable molecular biology equipment from actually wrote a blog post about the experiments that we were doing in Spain. I want to end the talk today by dispelling a myth. A lot of people think that being a scientist is a very lonely endeavour, that you spend a lot of time on your own pondering deeply scientific problems, and actually the reality couldn't be further from that. Science is a very, very collaborative endeavour, and every day I'm interacting with the people in my lab. So this is undergraduate students, master's students, PhD students, but also the people who work with me. I interact with other scientists within the school and the faculty. I collaborate with scientists in the UK and internationally. I still collaborate with scientists in New Zealand. So science is not only a collaborative endeavour, it's also an international endeavour. And so I want to take this opportunity to thank some of the people in my lab, and I've put a photograph here with some of the people in my lab who contributed to the work that I talked about today. So, we work together as a lab to really answer these scientific questions that we're interested in. And so most of my work that I've talked about today is about understanding gene-environment interactions, but also we're interested in the evolution of eusociality. So why is it that workers give up reproduction to support the reproduction of their queen? We also study ecosystem services and pollination networks and disease transfer in pollinator species and behavioural plasticity. And to do this, we use a variety of different tools, including model organisms, and I haven't talked about it today, but we use the fruit fly quite a lot, and we use molecular tools as well as behavioural assays to try and answer the questions that we're interested in. We also do a little bit of work using new technologies for passive monitoring of biodiversity. Finally, I just want to say thank you for listening to my biology taster lecture today, and have a great day.